You've heard about DNA, the double helix, the blueprint of life. What you probably haven't heard is why every single DNA polymerase on Earth can only copy in one direction, 5' to 3', never the reverse. This isn't some random quirk. This is chemistry making choices before the first cell ever existed, before bacteria, before anything alive. And when you see what this one restriction forces your cells to do just to copy your genome, you'll realize the machinery inside you is stranger and more desperate than anyone tells you. 3.6 billion years ago, when replication started, it locked into a pattern. No going back, no 3' to 5' synthesis, ever. Why? What makes the chemistry so stubborn? And what's the real cost of this directionality that's baked into every living thing? Most people think DNA replication means 2 strands unzip and both get copied smoothly. Wrong. One strand gets copied continuously. The other gets chopped into thousands of fragments stitched together later. That's happening in your cells right now. Your body synthesizes about 50 nucleotides per second. Bacteria hit 1,000 nucleotides per second. That's 16,000 letters added per minute in Escherichia coli. 42 minutes to duplicate 4.6 million base pairs. One circular chromosome. Yet this frantic speed still obeys the same directional law that started before oxygen filled the atmosphere. The answer sits in a single chemical reaction. DNA polymerase grabs a deoxynucleoside triphosphate. 3 phosphates hanging off the 5' carbon. The 3' hydroxyl group on the growing strand attacks the alpha phosphate. Nucleophilic attack. The beta and gamma phosphates snap off as pyrophosphate. Energy released. New phosphodiester bond formed. The chain grows 5' to 3' because that's where the energy comes from. The incoming nucleotide carries the fuel, not the strand. Switch that around and the whole system collapses. Why can't it go the other way? In theory, you could have 3' triphosphates on the incoming nucleotide. The 5' hydroxyl attacks. But that setup has a fatal flaw. Those triphosphates on the growing chain would sit there exposed to water. Spontaneous hydrolysis. In minutes, before the polymerase ever shows up, the activation energy is gone. No fuel, no synthesis. That's why every polymerase evolved with 5' triphosphates on the free nucleotides. Stable in solution, the 3' hydroxyl stays unreactive until the enzyme positions everything. 2 magnesium ions, precise geometry. Then the attack happens. One direction only. If you value documented molecular biology and evolutionary biochemistry like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual, using sources from the National Center for Biotechnology Information's Molecular Biology of the Cell, published by Garland Science, showing DNA polymerase mechanisms across all 3 domains of life. And from proceedings of the National Academy of Sciences USA, where Reiji and Tsuneko Okazaki published their 1968 discovery of discontinuous replication, describing short DNA fragments now bearing their name, documented through radioactive thymidine pulse labeling in Escherichia coli, showing fragments 1,000 to 2,000 nucleotides long in bacteria. Now back to the information. This directionality creates an immediate problem. DNA is anti-parallel. One strand runs 5' to 3', the other runs 3' to 5'. Helicase unzips them. Replication fork opens. Now you have 2 template strands facing opposite directions. DNA polymerase can only synthesize 5' to 3'. One template strand, the leading strand, lines up perfectly. Polymerase follows the fork, continuous synthesis, smooth, fast. The other strand, the lagging strand, points the wrong way. The fork moves away from the direction polymerase can work. What does the cell do? It cheats. Primacy drops an RNA primer. DNA polymerase extends it backward from the fork. Stops. Primase drops another primer. Polymerase extends again. Each fragment runs maybe 1,000 nucleotides in bacteria, 100 to 200 nucleotides in humans. These are Okazaki fragments, discovered February 1968. Reiji Okazaki and Tsuneko Okazaki at Nagoya University in Japan. They added radioactive hydrogen 3-thymidine to dividing Escherichia coli for 6 seconds. Pulse labeling. Then they lysed the cells under alkaline conditions. Alkaline sucrose gradient centrifugation. The newly synthesized DNA migrated near the top. Short fragments, not long continuous strands. They called it discontinuous replication. Reiji presented the findings at the Cold Spring Harbor Symposium in June 1968. Rollin Hotchkiss coined the term Okazaki pieces during that meeting, later shortened to Okazaki fragments. The discovery shattered the simple model where both strands grow continuously. Instead, the lagging strand is a patchwork. RNA primers everywhere. DNA polymerase fills between them, then another enzyme, DNA polymerase 1 in bacteria, chews off the RNA with its 5' to 3' exonuclease activity, replaces RNA with DNA, DNA ligase seals the Nix, phosphodiester bond formed between 3' hydroxyl and 5' phosphate. Finally, one continuous strand, but it took thousands of separate reactions to get there. Why does this matter? Because proofreading DNA polymerase 3 in Escherichia coli has a 3' to 5' exonuclease built in, the epsilon subunit. It checks each nucleotide after it's added. Wrong base pair, the polymerase backs up. Exonuclease clips it off. Polymerase tries again. This happens in the opposite direction from synthesis. 3' to 5' exonuclease chewing back toward the start. 5' to 3' polymerase building forward. That's the whole game. The energy for adding nucleotides sits on the incoming triphosphate. The 3' hydroxyl stays available for the next addition. If synthesis went 3' to 5', the energy would be on the growing strand. Spontaneous hydrolysis would wreck it. Proofreading would destroy the triphosphate activation. You'd clip off a wrong base and have no way to replace it. No fuel left. Synthesis stops. That's why no organism has ever evolved a 3' to 5' DNA polymerase. The chemistry forbids it, but the cost is real. Every time your cells copy DNA during S phase, which takes about 10 hours in many human cell types, the lagging strand gets synthesized in fragments. Your genome is 3 billion base pairs. That's 3 billion nucleotides per strand, 6 billion total. At 50 nucleotides per second, one polymerase would take over 690 days to finish. You'd never divide. Instead, you have 20,000 to 60,000 replication origins. Each origin fires. 2 replication forks move in opposite directions. Multiple polymerases work simultaneously. Leading strand synthesis, continuous. Lagging strand discontinuous everywhere. Polymerase delta handles the lagging strand in humans. Polymerase epsilon handles the leading strand. Both have 3' to 5' exonuclease activity. Proofreading. They catch errors immediately. Roughly 1 mistake per 100 million nucleotides before proofreading. After proofreading, 1 mistake per 1 billion nucleotides. Then mismatch repair proteins patrol behind them. MSH2. MSH6 proteins in humans. They scan for distortions in the helix. Wrong base pairs create bulges. Mismatch repair cuts out the error. DNA polymerase delta fills the gap. Ligase seals it. Final error rate drops to 1 mistake per 10 billion nucleotides. That's the target. Miss that, and you get cancer. In bacteria, the numbers are different, but the principle identical. DNA polymerase 3 has the alpha subunit for synthesis. The epsilon subunit for 3' to 5' exonuclease proofreading. The theta subunit stabilizes epsilon. Studies from Nagoya University in the 1990s through 2017 documented how Tsuneko Okazaki continued after Reiji died in August 1975 from leukemia. He was 44, born in Hiroshima. Exposed to radiation from the atomic bomb detonation August 6, 1945. 20 years later, he was researching DNA replication. 7 years after the Okazaki fragment discovery, he was gone. Tsuneko kept the lab running. She became the first female full professor at Nagoya University in 1983. She discovered that Okazaki fragments start with RNA primers, short stretches, 3 to 10 nucleotides. Later studies showed these primers are synthesized by primase, an RNA polymerase that can start synthesis de novo, no primer needed. Then DNA polymerase takes over. The RNA primer sits at the 5' end of each Okazaki fragment. DNA polymerase 1 in Escherichia coli has a 5' to 3' exonuclease in addition to its polymerase activity. It slides along behind DNA polymerase 3. Clips off ribonucleotides one at a time from the 5' end of the previous Okazaki fragment. Simultaneously adds deoxyribonucleotides to fill the gap. When it reaches the 3' hydroxyl of the next fragment, DNA ligase takes over. Ligase uses ATP in bacteria, NAD in some species, activates the 5' phosphate, 3' hydroxyl attacks, phosphodiester bond formed, one continuous strand, but trace the history and you see thousands of independent starts, thousands of ligation events, all because synthesis only goes one direction. Why didn't evolution find a different solution? Because it couldn't. The chemistry of phosphodiester bond formation requires energy input. That energy has to come from somewhere stable. Nucleoside triphosphates with 3 phosphates on the 5' carbon are stable in water for hours, even days. Nucleoside triphosphates with 3 phosphates on the 3' carbon would hydrolyze from the adjacent 2' hydroxyl in RNA, or would still face spontaneous breakdown in DNA. The 5' position is the only viable location. That fixes the direction. 3' hydroxyl attacks 5' alpha phosphate, chain grows 5' to 3'. No exceptions across 3 domains of life. Bacteria, archaea, eukarya, same mechanism, same direction, same problem. Some researchers asked if you could evolve a 3' to 5' polymerase. Theory says you'd need the triphosphate on the growing strand, head growth instead of tail growth. A few non-replicative polymerases use head growth for other reactions, but not DNA replication. Why? Because proofreading. A replicative polymerase has to copy accurately. Proofreading exonucleus must remove wrong bases. If the triphosphate is on the growing chain and you remove the terminal nucleotide, you clip off the activation energy for the next addition. Synthesis halts. To continue, you'd need to add a new triphosphate to the growing strand. That's a separate enzymatic step. Expensive, slow, error-prone. Natural selection crushed any lineage that tried. Only 5' to 3' polymerases survived. Only tail growth chemistry. Only one direction. The directionality also determines replication fork structure. At each fork, you have continuous synthesis on the leading strand and discontinuous synthesis on the lagging strand. The 2 polymerases don't work independently. They're part of a replisome complex. The replisome is a molecular machine. In Escherichia coli, it includes DNA polymerase 3 holoenzyme, helicase, primase, single-strand binding proteins, sliding clamp proteins, and more. Studies using electron microscopy in the 1970s and 80s showed the lagging strand loops out from the replisome. The trombone model, proposed by Bruce Alberts in 1980, the lagging strand template forms a loop. This orients both leading and lagging strand templates in the same direction relative to the replisome. Both polymerases move together. When the lagging strand polymerase completes an Okazaki fragment and hits the previous RNA primer, it releases. The loop collapses. A new primer is laid down. The loop reforms. Synthesis continues. The loop grows to about 1,000 nucleotides in bacteria, 100 to 200 in eukaryotes. Then the cycle repeats. This looping mechanism was confirmed by single-molecule experiments in the 2000s. Researchers attached fluorescent beads to DNA polymerase subunits, watched them move in real time. The leading strand polymerase moved steadily. The lagging strand polymerase paused every time it finished a fragment, released, rebound, paused again. The coordination between the 2 polymerases prevents the replication fork from stalling. If the lagging strand falls behind, the leading strand slows. If the leading strand outruns the lagging, the fork destabilizes. The replisome holds them together. Processivity. The term means how many nucleotides a polymerase adds before falling off. DNA polymerase 3 in Escherichia coli has processivity over 500,000 nucleotides when bound to the beta-sliding clamp. That clamp is a ring-shaped protein. It encircles the DNA, tethers the polymerase, prevents dissociation. Without the clamp, processivity drops to 10 or 20 nucleotides, useless for replication. The sliding clamp is loaded onto DNA by a clamp loader complex. The clamp loader is an ATPase. It uses ATP hydrolysis to open the clamp ring. Slides it onto the DNA at a primer template junction. Closes the ring. The polymerase binds the clamp. Synthesis starts. When synthesis finishes, the clamp must be removed. Otherwise, it blocks other enzymes. In bacteria, the clamp loader also removes the clamp. In eukaryotes, different proteins handle clamp loading and removal. pCNA is the eukaryotic sliding clamp, proliferating cell nuclear antigen. It's a trimer. 3 subunits form a ring. RFC is the clamp loader. Replication factor, C, 5 subunits. RFC loads pCNA onto primed DNA. Polymerase delta or epsilon binds pCNA. Synthesis proceeds after the Okazaki fragment is complete. pCNA slides off or gets recycled for the next fragment. Eukaryotic cells have another layer of complexity. Chromatin, DNA wrapped around histone, octamers, nucleosomes every 200 base pairs on average. Ahead of the replication fork. Nucleosomes must be disrupted. Behind the fork, they must be reassembled. Histone chaperones manage this. They peel histones off the parental DNA, hold them temporarily, deposit them onto the new daughter strands. Some parental histones are retained. Some new histones are synthesized. Histone H3, variant H 3.3 gets incorporated. Epigenetic marks on histones can be inherited or reset. This adds another regulatory layer. But the core replication mechanism remains the same: 5' to 3' synthesis, leading strand continuous, lagging strand discontinuous. The evolutionary constraints run deep. The RNA world hypothesis suggests that early life used RNA genomes. RNA polymerases can initiate synthesis de novo. No primer required, but RNA is chemically unstable compared to DNA. The 2' hydroxyl makes RNA prone to hydrolysis. Base-catalyzed cleavage. DNA lacks that hydroxyl, more stable, better for long-term information storage. When the transition from RNA to DNA happened, the replication machinery had to adapt. DNA polymerases evolved from RNA polymerases. But DNA polymerases lost the ability to start de novo. They require primer. Why? Because fidelity. Starting de novo is error-prone. The first few nucleotides are often mismatched. A primer provides a stable base, higher accuracy, but it means you need a separate enzyme, primase, to lay down RNA primers. Then DNA polymerase extends them. Then another enzyme removes the RNA. Then ligase seals the gaps. 5 separate enzymatic steps just to handle the lagging strand. All because DNA polymerases can't start de novo and can't synthesize 3' to 5'. Is there any organism that breaks this rule? No. A few specialized polymerases have been discovered with unusual properties. The Th1 superfamily enzymes can add nucleotides to the 5' end of tRNA in a 3' to 5' direction. But these are not replicative polymerases. They're editing enzymes. They repair specific tRNA molecules. The mechanism is different. They don't use the same 2-metal ion catalysis that replicative polymerases use. They're evolutionary outliers. They don't replicate genomes. For chromosomal DNA replication across all known life, the direction is always 5' to 3'. This universality suggests the mechanism was fixed early, before the last universal common ancestor, LUCA, somewhere between 3.8 and 4.1 billion years ago. The chemical logic locked in. Once a functional replication system existed, changing the direction would require redesigning the entire machinery. Polymerase active site, proofreading exonucleus, primase, ligase, sliding clamp, helicase. Topoisomerase, all coordinated, all dependent on 5' to 3' synthesis. A mutation that tried to reverse the direction would be lethal. No intermediate state is viable. It's a frozen accident. But it's a frozen accident driven by chemistry, not chance. What does this directionality cost in terms of energy and time? Every Okazaki fragment requires an RNA primer. Primase synthesizes that primer. Each primer is 3 to 10 ribonucleotides. Primase doesn't proofread. Error rate is high, about 1 mistake per 1,000 to 10,000 nucleotides, but the primers are temporary. They get removed, so the errors don't matter long term. Still, synthesizing and removing primers costs ATP and GTP, ribonucleotide triphosphates for primer synthesis. Then DNA polymerase 1, or RNase H, removes the RNA, uses its exonucleus activity. Each cleavage releases a nucleoside monophosphate. Then DNA polymerase fills the gap, uses deoxyribonucleotide triphosphates. Each addition releases pyrophosphate. Pyrophosphahase hydrolyzes pyrophosphate to 2 phosphates. Drives the reaction forward, but it means you're spending extra nucleotide triphosphates on the lagging strand compared to the leading strand. In Escherichia coli, replicating 4.6 million base pairs requires synthesizing about 4,600 Okazaki fragments on the lagging strand, assuming 1,000 nucleotides per fragment. Each fragment needs 1 RNA primer. That's 4,600 primers. Each primer is maybe 5 nucleotides on average, 23,000 ribonucleotides. Then you remove them, 23,000 deoxyribonucleotides to replace them, plus 4,600 ligation events. Each ligation uses 1 ATP in Escherichia coli, so you're spending at least 4,600 ATP just on ligation. Add the cost of primer synthesis and removal. And the lagging strand is significantly more expensive than the leading strand, but the cell pays that cost because there's no alternative. In humans, the cost scales up. 3 billion base pairs per haploid genome. 6 billion for both strands. Estimate 150 nucleotides per Okazaki fragment. That's 20 million fragments per replication cycle, 20 million RNA primers synthesized and removed, 20 million ligation events. Each uses 1 ATP. 20 million ATP just for ligation on the lagging strand. That's on top of the 6 billion deoxyribonucleotide triphosphates needed for synthesis. And this happens every time a cell divides. Your body has roughly 37 trillion cells. Most of them don't divide frequently, but in tissues with high turnover, like the gut lining or bone marrow, cells divide constantly. Billions of replications per day. Trillions of Okazaki fragments synthesized and stitched together, all because DNA polymerase can only work in one direction. If you value deep molecular research like this, subscribe. It's the best way to support the channel. Comment where you're watching from. This script uses research from Nature Scientific Reports published June 2020, showing evolutionary models where anti-parallel DNA structure enables parallelized replication by dividing the genome into independently replicating segments. And from PNAS, published February 2019, solving the 50-year-old Okazaki fragment problem by demonstrating that ribonucleotide incorporation into the leading strand by DNA polymerase epsilon creates short fragments that were previously misattributed entirely to lagging strand synthesis. Now back to the information. The directionality also determines the structure of the replication machinery. DNA polymerase has a palm domain that holds the active site, a finger's domain that binds the incoming nucleotide, a thumb domain that grips the DNA. The active site contains two magnesium ions, magnesium A and magnesium B. They coordinate the 3' hydroxyl and the triphosphate, lower the activation energy for the nucleophilic attack. Without the magnesium ions, the reaction is too slow. With them, the reaction rate is around 250 nucleotides per second for the epsilon subunit proofreading exonuclease in Escherichia coli. The polymerase itself, DNA polymerase 3, alpha subunit Y, synthesizes at 1,000 nucleotides per second. That's fast, but proofreading slows it down slightly. The overall rate is 500 to 1,000 nucleotides per second, depending on conditions. In eukaryotes, polymerase epsilon and delta are slower. Around 40 base pairs per second per fork for Drosophila melanogaster replication measured in vivo. In humans, replication forks move at roughly 1.5 kilobases per minute on average. That's 25 nucleotides per second per fork. Much slower than bacteria. Why? Chromatin. Nucleosomes slow down the fork. Histone chaperones need time to disassemble and reassemble nucleosomes. Also, eukaryotic polymerases have more extensive proofreading, more protein-protein interactions, more regulatory checkpoints. The replication machinery is larger. More subunits, more coordination. That trades speed for accuracy and control. But the fundamental chemistry is identical. 3' hydroxyl attacks 5' alpha phosphate, 5' to 3' synthesis. The proofreading mechanism deserves closer attention. When DNA polymerase adds a nucleotide, it checks the geometry of the base pair. Correct base pairs, AT and GC, fit into the active site. The Watson-Crick geometry is recognized. The finger's domain closes around the incoming nucleotide. The polymerase undergoes a conformational change. The bond forms, the fingers open. The polymerase translocates one nucleotide forward. If an incorrect nucleotide is added, the geometry is wrong. The mismatch distorts the helix. The polymerase stalls. The primer terminus with the mismatched base phrase, single-stranded. It swings out of the polymerase active site into the exonuclease active site. The exonuclease, 3' to 5' bias, clips off the mismatched nucleotide. The primer terminus swings back into the polymerase site. The polymerase tries again, adds the correct nucleotide. Synthesis continues. This process is called kinetic proofreading. The incorrect nucleotide is less stable in the active site, more likely to dissociate before the bond forms. And if the bond does form, the mismatch is more likely to be excised by the exonuclease. The combination of selectivity during insertion and selectivity during excision reduces the error rate by a factor of 100 to 1,000. But it only works if the exonuclease operates in the 3' to 5' direction. Opposite to synthesis, if synthesis and excision went in the same direction, the polymerase would just chew back everything it synthesized. No net progress. The anti-parallel directionality is essential for proofreading. What happens when proofreading fails? Mismatch repair. In Escherichia coli, the MATES protein recognizes mismatches. It binds the distorted helix. Recruits MATL. Together, they activate NUT-H, an endonuclease that cuts the newly synthesized strand. The cut is made near a hemimethylated GATC sequence. These sequences get methylated by DAM methylase shortly after replication. But there's a delay, a few minutes. During that window, the new strand is unmethylated. The parental strand is methylated. Much cuts the unmethylated strand. Exonucleases degrade the DNA from the nick toward the mismatch DNA polymerase. 3 resynthesizes the degraded region. DNA ligase seals the nick. The mismatch is gone. In eukaryotes, mismatch repair is similar but uses different proteins. MSH2 and MSH6 form a complex that recognizes mismatches. MSH2 and MSH3 recognize insertion deletion loops. These complexes recruit MLH1 and PMS2. They coordinate excision and resynthesis. The newly synthesized strand is identified by nicks left by Okazaki fragment processing on the lagging strand, or by polymerase epsilon on the leading strand. Exonuclease 1, a 5' to 3' exonuclease, degrades from the nick. Polymerase delta fills the gap. Legacy seals it. Error rate drops another factor of 100 to 1000. When mismatch repair is defective, mutation rates skyrocket. In humans, loss of mismatch repair genes like MSH2 or MLH1 causes Lynch syndrome. Hereditary non-polyposis, colorectal cancer. Tumors accumulate mutations 100 times faster than normal. Microsatellite instability. Short repetitive sequences expand and contract. Frame shifts, protein truncations, oncogenes activated, tumor suppressors lost, cancer. The same happens if proofreading fails. Mice with a proofreading deficient polymerase delta develop tumors. The exonuclease domain has a point mutation. Aspartate to alanine, the catalytic activity is gone. The mouse survives to birth, but develops cancer within months. Colon tumors, lymphomas. The mutation rate is 700 to 8,000 times higher than wild type. An error catastrophe at the organismal level. This demonstrates the evolutionary pressure to maintain replication fidelity. Error rates above a certain threshold are incompatible with complex life. The threshold is roughly 1 mistake per 10 million nucleotides per generation for a mammalian genome. Below that, selection can purge deleterious mutations. Above that, mutations accumulate faster than selection can remove them. Muller's ratchet. The genome degenerates. Extinction. DNA polymerase directionality, proofreading, and mismatch repair are the mechanisms that keep error rates below the threshold. Without them, no mammals, no birds, no flowering plants, no complex multicellular life. Now consider the implications for the origin of life. RNA genomes came first, RNA polymerases, RNA replication. But RNA is chemically unstable. The transition to DNA required DNA polymerases. Those polymerases had to maintain fidelity. Proofreading evolved, 3' to 5' exonuclease, but that only works if synthesis goes 5' to 3'. If early polymerases tried both directions, the proofreading competent version won, natural selection, billions of generations. Only the 5' to 3' lineages survived. Once that directionality was established, it became universal. Every descendant inherited it. Every replication fork obeys it. Every cell in your body depends on it. What about viruses? Do they follow the same rule? Yes. DNA viruses use host DNA polymerases or encode their own. Bacteriophage, T4 encodes DNA polymerase, gene 43 product. It has polymerase activity and 3' to 5' exonuclease activity. Proofreading synthesizes 5' to 3'. Okazaki fragments on the lagging strand. Same mechanism. Herpes simplex virus encodes its own polymerase. UL30 gene product, proofreading exonuclease, 5' to 3' synthesis. Adenovirus has a different strategy. It uses protein priming instead of RNA priming. A terminal protein binds the 5' end of the genome. The polymerase adds nucleotides to a serine or threonine residue on the protein, then extends continuously. No Okazaki fragments. But the polymerase still works 5' to 3'. Continuous synthesis on both strands is possible because the ends of the linear genome are bound by terminal proteins. No free 5' ends, but the chemistry is identical. 3' hydroxyl attacks, 5' alpha phosphate. RNA viruses are different. They use RNA-dependent RNA polymerases. These enzymes can initiate de novo, no primer required, but they still synthesize 5' to 3'. The incoming ribonucleoside triphosphate has 3 phosphates on the 5' carbon. The 3' hydroxyl on the growing strand attacks the alpha phosphate. Same chemistry, same direction. Some RNA viruses, like influenza, use a cap snatching mechanism. They steal the 5' cap from host mRNAs, use it as a primer, then extend with viral RNA polymerase. But again, 5' to 3' synthesis. Retroviruses reverse transcribe RNA into DNA. Reverse transcriptase is an RNA-dependent DNA polymerase. It synthesizes DNA from an RNA template, still 5' to 3'. uses a tRNA primer, extends the primer, synthesizes the first DNA strand, then degrades the RNA template with RNase H activity built into the same protein, synthesizes the second DNA strand. Both strands are made 5' to 3', discontinuous synthesis on one strand, continuous on the other. The final product is double-stranded DNA with long terminal repeats, integrated into the host genome. But the replication chemistry is standard: 5' to 3' synthesis, 3' hydroxyl attacks, 5' alpha phosphate. Even telomerase, the enzyme that extends chromosome ends in eukaryotes, works 5' to 3'. Telomerase is a reverse transcriptase. It carries an RNA template. The RNA template has a sequence complementary to the telomere repeat. In humans, the telomere repeat is T-tag-GS. The RNA template is AAUC-CC repeated. Telomerase binds the 3' overhang of the chromosome, extends it by adding T-tag-G repeats, 5' to 3' synthesis. The 3' hydroxyl of the chromosome attacks the 5' alpha phosphate of the incoming deoxyribonucleoside triphosphate. 6 nucleotides added. The enzyme translocates, adds another repeat, extends the 3' strand. Then DNA polymerase alpha primase synthesizes the complementary 5' to 3' strand on the other template. The chromosome end is extended. This happens in germ cells, stem cells, and most cancer cells. In somatic cells, telomerase is repressed. Telomeres shorten with each division, Hayflick limit. The cell senesces after 50 to 70 divisions. The directionality also determines how replication terminates. In Escherichia coli, CO, the circular chromosome has a termination region opposite the origin. Tear sequences, to its protein binds, tear sequences, blocks helicase in one direction, allows it in the other. The two replication forks meet in the termination region. DNA synthesis completes. The two circular chromosomes are catenated, interlocked. Topoisomerase 4 introduces double-strand breaks, passes one chromosome through the other, reseals the breaks, the chromosomes separate. Cell division proceeds. In eukaryotes, termination is more complex. Multiple replication origins, multiple forks. Forks from adjacent origins converge. Leading strand synthesis continues until the forks meet. The last Okazaki fragment on the lagging strand is synthesized. The RNA primer is removed. DNA polymerase delta fills the gap. DNA ligase seals the last nick. The chromosome is fully replicated, but linear chromosomes have a problem. The 5' ends cannot be fully replicated. The last RNA primer on the lagging strand leaves a gap. When the primer is removed, there is no upstream Okazaki fragment to fill the gap. The chromosome shortens. Telomerase solves this in cells where it's active. In cells without telomerase, the telomeres shorten until they reach a critical length. DNA damage response activates. Cell cycle arrest, senescence, or apoptosis. What does all this mean? DNA replication directionality is not a minor detail. It's a fundamental constraint that shapes the machinery, the cost, the speed, and the fidelity of genome duplication. The 5' to 3' synthesis direction is locked in by chemistry. The instability of 3' triphosphates, the requirement for proofreading, the impossibility of continuous synthesis on both strands. These constraints force cells to use Okazaki fragments, RNA primers, ligase, thousands of extra enzymatic steps, millions of extra ATP molecules consumed. All because the 3' hydroxyl can only attack the 5' alpha phosphate in a stable system. The Okazaki fragments themselves are a monument to this constraint. Reiji and Tsuneko. Okazaki discovered them in 1968. Their experiments showed that lagging strand synthesis is discontinuous. Short fragments, pulse-labeled with radioactive thymidine, detected by alkaline sucrose gradient centrifugation. The discovery was published in Proceedings of the National Academy of Sciences USA in February 1968. Volume 59, Issue 2, pages 598 to 605. The title: Mechanism of DNA chain growth. 1. Possible discontinuity and unusual secondary structure of newly synthesized chains. Authors: R. Okazaki, T. Okazaki, K. Sakabe, K. Sugimoto, Asugino. They used Escherichia coli, added hydrogen-3 thymidine for short pulses, analyzed the DNA by sedimentation, found small fragments, 1,000 to 2,000 nucleotides long. Later experiments showed these fragments contain RNA at the 5' end. Primase synthesizes the RNA. DNA polymerase extends it. RNase H, or DNA polymerase 1, removes the RNA. DNA ligase joins the fragments. Tsuneko. Okazaki continued the work after Reiji died. She identified the RNA primers, showed they are 3 to 10 nucleotides long, demonstrated they are synthesized by primase, proved the primers are at the 5' end of Okazaki fragments. This work was published in Cold Spring Harbor Symposia on Quantitative Biology in 1979, volume 43. Pages 203 to 219. Title: Structure and Metabolism of the RNA Primer in Discontinuous Replication of Prokaryotic DNA. Authors: T. Okazaki, Y. Kurosawa, T. Ogawa, T. Seki, K. Shinozaki, S. Hirose, of Fujiyama, I. Kohara, Y. Machida, F. Tamanoi, T. Hozumi. They used temperature-sensitive mutants of bacteriophage T4, gene 30 mutant, defective in DNA ligase at the high temperature. Okazaki fragments accumulate. Fragments can be isolated, analyzed. RNA primers detected, sequenced. The discovery of Okazaki fragments solved a 50-year mystery. How can both strands be synthesized simultaneously if DNA polymerase only works in one direction? Answer: one strand is continuous, the other is discontinuous. The leading strand extends smoothly. The lagging strand is chopped into fragments and stitched together later. This model has been confirmed by decades of research: single molecule studies, electron microscopy, biochemical reconstitution, genetic analysis, crystal structures of polymerases. Everything fits. 5' to 3' synthesis, Okazaki fragments on the lagging strand, RNA primers, proofreading exonuclease, 3' to 5'. Mismatch repair, fidelity maintained, genome copied. Cell divides. A recent twist came in 2019, a paper in PNAS by Cronin, Kuzmanova, and Kuzmanov, published February 2019, volume 116, that's new. Pages 1251 to 1259. Title: Near continuously synthesized leading strands in Escherichia. Coli are broken by ribonucleotide excision. They showed that even the leading strand has small fragments. Not classic Okazaki fragments, but short stretches where ribonucleotides get incorporated. DNA polymerase epsilon occasionally inserts a ribonucleotide instead of a deoxyribonucleotide. About 1 ribonucleotide per kilobase in yeast. RNase H2 recognizes the ribonucleotide, cuts the DNA backbone, creates a nick. The nick is processed. Strand is resynthesized, ligated, so even the leading strand isn't perfectly continuous. It has embedded ribonucleotides that get excised and repaired, but the scale is different. 1 ribonucleotide per 1,000 nucleotides on the leading strand. 1 Okazaki fragment per 100 to 1,000 nucleotides on the lagging strand. The leading strand is still much more continuous, but not absolutely continuous. This finding resolved a long-standing puzzle. Pulse label experiments in the 1960s and 70s found small DNA fragments on both strands. Researchers assumed both were Okazaki fragments, but later work showed the leading strand should be continuous. The 2019 paper reconciled the discrepancy. Leading strand fragments come from ribonucleotide excision repair, not Okazaki fragments, but they show up in pulse label experiments because the labeling time is short, a few seconds. In that time, nicks from ribonucleotide excision haven't been sealed yet, so the leading strand DNA is fragmented. But after longer chase times, the nicks are sealed. The leading strand is continuous. The lagging strand remains fragmented longer because Okazaki fragment processing takes more time. What does this tell us about the evolutionary optimization of DNA replication? The system is a compromise. 5' to 3' synthesis is mandatory. Chemistry dictates it. Proofreading requires it, but that forces discontinuous synthesis on one strand. Cells have adapted. Primus lays down primers. DNA polymerase extends them. Exonuclease removes the primers. Ligase seals the gaps. The machinery is elaborate, coordinated, fast, accurate, but it's built around a fundamental asymmetry. The 2 strands cannot be treated identically. One is easy, the other is hard. The cost is paid in enzymes, ATP, and time, but the benefit is fidelity. Error rates low enough to sustain complex genomes, low enough to prevent error catastrophe, low enough to allow evolution to explore sequence space without collapsing into mutational meltdown. The final piece of this puzzle is the evolutionary constraint. Once the 5' to 3' synthesis direction was established, it became irreversible. Too many systems depend on it. Polymerase structure, exonuclease orientation, primer requirements, sliding clamp loading, replication fork architecture, topoisomerase action, telomere replication, Every aspect of DNA metabolism assumes 5' to 3' synthesis. Changing it would require redesigning everything simultaneously. No intermediate state is viable. A partially functional replication system is a dead cell. Natural selection cannot bridge that gap. The direction is locked. This is the answer. DNA only copies in one direction because the chemistry of phosphodiester bond formation requires stable triphosphates on the incoming nucleotides. 3' triphosphates are unstable. 5' triphosphates are stable. The 3' hydroxyl attacks the 5' alpha phosphate. The chain grows 5' to 3'. Proofreading requires a 3' to 5' exonuclease operating in the opposite direction. Both must coexist. That asymmetry forces discontinuous synthesis on the lagging strand, Okazaki fragments, RNA primers, Legacy. Thousands of extra steps. Millions of extra ATP spent, but no alternative exists within the constraints of chemistry and evolution. Every replication fork in your body right now obeys this rule. Every bacteria, every virus, every living thing, the direction was set before the first cell, before the first chromosome, before oxygen, before photosynthesis. When RNA polymerases first started copying templates, the chemistry chose 5' to 3'. When DNA polymerases evolved, they inherited the same constraint. 3.6 billion years later, the pattern persists, unchanged, unchallengeable. In 2020, meantime, a paper in Scientific Reports examined the evolutionary advantage of antiparallel DNA strand orientation. Authors, Kumar and Rao, published June 2020. They argued that antiparallel strands enable parallelized replication by dividing the genome into independently replicating segments. Okazaki fragments can be synthesized simultaneously on the lagging strand, while the leading strand proceeds continuously. This doubles the effective replication rate compared to sequential copying of the entire genome. Natural selection favored antiparallel structure because it allows faster replication. Faster replication means faster division, faster growth, competitive advantage. The organisms that optimized replication speed outcompeted slower replicators. The 5' to 3' directionality, combined with antiparallel strand orientation, became universal. So when you look at DNA replication, you're seeing chemistry making choices. The 3' hydroxyl group on deoxyribose is a weak nucleophile. It needs activation. 2 magnesium ions in the polymerase. Active site coordinate the reactants, lower the activation energy. The alpha phosphate on the incoming nucleotide is electrophilic. The 3' oxygen attacks the phosphorus, pentacovalent transition state. The beta and gamma phosphates leave as pyrophosphate. The phosphodiester bond forms. The reaction is favorable because pyrophosphate hydrolysis is highly exergonic. Negative delta G. 2 phosphate molecules released. Each hydrolysis releases about 7 kilocalories per mole, drives the reaction forward. But this mechanism only works if the triphosphate is on the incoming nucleotide and the hydroxyl is on the growing chain. Reverse it, and the system fails. At a human replication fork, moving at 50 nucleotides per second, the machinery adds 1 nucleotide every 20 milliseconds. In that time, the polymerase has to select the correct DNTP from the pool, bind it, check the base pairing, close the fingers domain, position the magnesium ions, catalyze the reaction, open the fingers, translocate 1 nucleotide forward, release pyrophosphate, check for errors. If an error is detected, transfer the primer terminus to the exonuclease site, clip off the wrong base. Transfer back to the polymerase site, try again, all in 20 milliseconds. The coordination is extraordinary. The speed is extreme. The accuracy is astonishing. 1 mistake per billion nucleotides, that's 9 zeros. 1 in 1 with 9 zeros after it, achieved by a molecular machine made of proteins, no consciousness, no intent, just chemistry and structure and evolution. The last detail, where does this leave us? DNA replication directionality is a constraint that defines life. It shapes genomes. It determines mutation rates. It influences evolution. It makes cancer possible when it fails. It enables complex organisms when it succeeds. The 5' to 3' direction is written into the structure of nucleotides, the mechanism of polymerases, and the history of life. You can't change it without unmaking biology. A final revelation. In the termination region of Escherichia coli, where the 2 replication forks meet, there's a narrow zone where both forks have just finished synthesis. For a few seconds, both strands are fully copied. The RNA primers are gone. The Okazaki fragments are joined. The 2 circular chromosomes are complete, but catenated. Topoisomerase 4 hasn't separated them yet. In that moment, the genome exists as 2 interlocked circles. 42 minutes of replication compressed into a topological knot. Then topoisomerase 4 acts. Double strand break, pass through, reseal. The circles separate. The cell divides, and the whole process begins again. 5' to 3', leading strand continuous, lagging strand discontinuous, Okazaki fragments, primers, ligas, the cycle that has repeated since the origin of life, the direction that cannot change. The chemistry that built the world